Hello and welcome to the class today. We will be discussing carbohydrate chemistry. Carbohydrate chemistry. What are carbohydrates? Carbohydrates are aldehyde or ketone compounds with multiple hydroxyl groups. They are known to form the bulk of the organic matter on planet Earth. So you find carbohydrates in plants and animals. They are in fruits and vegetables. They form the bulk of the organic matter on planet Earth. And carbohydrates have elements like carbon, hydrogen, and oxygen in their structure. Some complex carbohydrates have nitrogen and sulfur also present in their structures. So, what are their roles or what are the functions of carbohydrates? Carbohydrates serve as a good energy store. They serve as good energy store in the form of starch or glycogen stored up in the liver, starch stored up in plants. They are known to form the structural framework of the hereditary material, DNA, and also they form the structural framework of ribonucleic acid, RNA. They are also structural elements in the cell walls of bacteria and plants. Carbohydrates are found as exoskeleton in insects. Examples of carbohydrates that are found in the exoskeleton of insects is chitin and cellulose are carbohydrates found on the cell wall. Carbohydrates are linked to proteins, and when they are linked to proteins, they are referred to as glycoproteins. When they are linked to lipids, they are referred to as glycolipids. And they help when they are glycoproteins or glycolipid, they help in cell-to-cell recognition processes. So a cell can easily be identified based on the type of glycoprotein or glycolipid that is found on the cell membrane. Classes of carbohydrates. We have several classes of carbohydrates. Basically, they are classified into four major groups. The monosaccharides, they are the simplest form of carbohydrates in they are the simplest form of carbohydrates that have been studied, and they contain aldehyde or keto groups in their structures. Examples of the monosaccharides include trioses, tetroses, pentoses, exoses. Trioses, a good example, is the glyceraldehyde. Tetroses, a good example is erythros. Pentoses, ribose sugar found in DNA or RNA, is a pentose sugar, and exoses, the most common of them all, is glucose. Carbohydrates can be classified as disaccharides, which are condensation products of two monosaccharide units. Example is sucrose, which is the normal table sugar we have in our homes, maltose, and lactose, milk sugar. Oligosaccharides are a class of carbohydrates that contain between 3 to 10 monosaccharide units. These monosaccharide units are linked by what is called a glycosidic bond. Example of an oligosaccharide is raffinose. Raffinose is an example of an oligosaccharide. Polysaccharides are a class of carbohydrates and they are polymers of sugars. They contain more than 10 monosaccharide units joined together by glycosidic linkage. Polysaccharides such as starch are found in plants. Polysaccharides such as glycogen are found in animal tissues, and cellulose is also a polysaccharide that makes up the cell wall of plants and some bacteria. Monosaccharides are the simplest form of carbohydrates, like we have earlier mentioned, and they have aldehyde or keto group attached to them, and they also have more than two or more hydroxy group. They have the empirical formula CH2O bracket N. So you want to calculate the formula of a specific monosaccharide, you use this formula, this empirical formula CH2O. and N equals to the number of carbon, which can range between 3 and 6. Now, monosaccharides have chiral centers, which are also known as asymmetric carbon, and the presence of chiral centers of this asymmetric carbon gives rise to many stereoisomers or enantiomers. And so you can see a monosaccharide, a compound occurring either in the L configuration or the D configuration. Glyceraldehyde is an aldol sugar, while dihydroxyacetone is a ketone sugar, and they are the smallest members of the monosaccharide family. D-glucose and D-fructose are the two most common exoses we find. Now, natural sugars are known to occur in the D configuration, except for arabinose, which occurs in the L configuration. And sugars that have more than one asymmetric carbon atom, that's more than one chiral centers, would also have symbols like D or L attached to them. The symbols D or L refer to the absolute configuration of the asymmetric carbon that is farthest from the aldehyde or keto group. The D or L symbol refer to the absolute configuration of the asymmetric carbon farthest from the aldehyde or keto group. And D or L shows that those sugars are optically active. Now, the number of stereoisomer a sugar will have is determined by the formula 2 to the power of N, where N is the number of chiral centers in that particular sugar. Two sugars that differ only in their configuration around one carbon are known as epimers. Example, mannose is a 2-epimer of glucose, and galactose is a 4-epimer of glucose, because the configuration of the hydroxy group and the hydrogen ion around the carbon in glucose at position 2 and 4 differ in galactose and in mannose. Two sugars that have opposite configuration at the same carbon atoms are called diastereoisomers. And so from the diagram here, we see the structure of glyceraldehyde and dihydroxyacetone, which are the smallest member of the monosaccharide unit. Glyceraldehyde is an aldol trial sugar, while dihydroxyacetone is a keto trial sugar. D-glucose is an aldol sugar, while D-fructose is a keto sugar. And you can see their structures. Now, this table or this diagram shows the structure of various monosaccharides. Glucose in its ring structure, fructose in its ring structure, and galactose. Now, if we look at the position of hydroxy group at position 4 of galactose and compare to that of glucose at position 4, we can see that the position of the hydroxy group differs for both sugar. It differs for both sugar. Now, this diagram shows the ball and stick model as well as the Fisher projection formula for D-glyceraldehyde and L-glyceraldehyde. The ball and stick model shows the mirror image of this particular compound, while the Fisher projection shows us the position of the hydroxy group around the same carbon. And you can see that the hydroxy group on this position of glyceraldehyde is in the D configuration, and the hydroxy group on the same carbon atom here is in the L configuration. Now, this diagram shows the structures of monosaccharides that have 6 carbons in their structure. They are all aldoses. We can see the structure of D-allose, D-altrose, D-glucose, D-mannose, D-glucose, D-idose, D-galactose, and D-tallose. The sugars are all in the D configuration. In aqueous solution, aldotetrosis and all monosaccharides with 5 or 6 carbon atoms forms ring structures. When they form ring structures, the carbonyl group forms a covalent bond with the oxygen of a hydroxy group along the chain. And when this happens, you have either a paranose formed or a furanose formed. Now this is D-glucose, and in solution forms a ring structure, and the carbonyl carbon here interacts with the hydroxy group here to form a complete ring structure. And then it forms either an alpha D glucoparanose or a beta D glucoparanose. The position of the hydroxy group here will determine whether it is an alpha D glucose or a beta D glucose. Here, the hydroxy group is below the plane, and here the hydroxy group is above the plane. So this is alpha D glucoparanose, and this is alpha D fructopyranose. For further understanding, this diagram shows a picture of a beta D glucopyranose, and this is the structure of a beta D fructopyranose. This ring structure here that glucose has is known as a paran ring, and that of fructose is known as a furan ring. We have some monosaccharides that are called derived monosaccharides. Example is sugar acid, example glucuronic acid, and in biochemistry, it is used for removal of toxic compound. We also have some sugar alcohols. Example is mannitol, that is derived from mannose, and ribitol derived from ribose. Amino sugars also exist as derived monosaccharides. Examples are, an example of such is galactosamine, found in the exoskeleton of insects, galactosamine. Now we look at disaccharides. A very good example of disaccharide is maltose, and it's derived from malt. Maltose is formed from 2 glucose units that are linked together by an alpha-1,4 glycosidic linkage. Maltose is a reducing sugar because their numeric carbon is free for interaction, for bond formation with some other molecules. Now, maltose yields only D-glucose units on hydrolysis. The enzyme responsible for cleaving maltose is known as maltase, and this enzyme is specific only for a glycosidic linkage. Now, these are structures of common disaccharides. This is lactose, made up of beta D-galactopyranose, linked by alpha-1 linkage to beta D-glucopyranose. And this is sucrose, beta D-fructofuranosyl, alpha D-glucopyranoside. And this is the structure of trehalose, alpha D-glucopyranose and alpha D-glucopyranoside. The linkage here. We can see the alpha beta linkage here, the beta linkage here. This is an alpha-alpha linkage in trehalose. Now, the structure here is that of alpha D-glucose interacting with beta D-glucose. If it's a forward reaction, it's known as condensation reaction, and it gives out a molecule of water to form maltose. Maltose, on the other hand, can be hydrolyzed to form alpha D-glucose and beta D-glucose. Isomaltose is also formed from two glucose units but are linked by a beta-1,6 glycosidic linkage. It is linked by a beta-1,6 glycosidic linkage, and it is a reducing sugar, meaning that it's an anomeric carbon. One of the sugars, the anomeric carbon, is free for reaction. Sucrose is known as cane sugar, is derived from sugar cane and contains glucose and fructose linked together by an alpha-1,2 glycosidic linkage. It is a non-reducing sugar because its anomeric carbon atom is not available for reaction for bond formation. Sucrose is a non-reducing sugar. Lactose, a disaccharide known as milk sugar, consists of galactose and glucose, and they are linked together by a beta-1,4 glycosidic bond. Cellulose is the simplest repeating unit of cellulose, and it contains two monosaccharide units of glucose which are linked together by a beta-1,4 glycosidic linkage. Oligosaccharides, on the other hand, are formed from 3 to 10 monosaccharide units of sugars come together. Examples of oligosaccharides, we have raffinose, stachyose, isomaltose, and pannose. They are found in several natural sources such as raffinose or babacose are found in beans, in peas, and other legumes. Kestose and neokestose are found in some fruits such as apples and grapes. Maltodextrose and isomaltose are found in some grains such as barley and wheat. Raffinose is a trisaccharide, and it has galactose, glucose, and fructose in its structure. Stachyose is a tetrasaccharide, and it is composed of two galactose units, one glucose unit, and one fructose unit. Pannose, on the other hand, is a trisaccharide composed of glucose, maltose, and glucose units. Kestose is composed of glucose, fructose, and glucose in its structure. Now, this is the structure of the oligosaccharide raffinose. It has galactose linked by, and this is linked by a 1,6 linkage to glucose, and then glucose 1,2 to fructose linkage. That's the structure of raffinose. Polysaccharides are carbohydrates made up of many monosaccharide units. They have high molecular weight and can be grouped as skeletal or structural polysaccharide. Example is cellulose and chitin. They can be grouped as nutrient polysaccharide, e.g. starch and glycogen. Now, depending on the monosaccharide unit present in these carbohydrates, polysaccharides can be grouped either as homopolysaccharides or heteropolysaccharides. Chitin is a principal component of the exoskeleton of arthropods like insects, lobsters, and crabs. And they have a linear homopolysaccharide of N-acetylglucosamine linked together in the beta-1,4 linkage. This is N-acetylglucosamine. At carbon atom number 2, there's an acetyl group attached instead of an hydroxyl group. And that's what you find here. So it's linear homopolysaccharide containing N-acetylglucosamine linked to the N-acetylglucosamine in a beta-1,4 linkage. And it's found as exoskeleton of insects. Glycogen is a stored form of polysaccharide in animals and is found abundantly in the liver and very little is found in the skeletal muscle. It has long straight chains linked by alpha 1,4 glycosidic linkage and branches at alpha 1,6 glycosidic bonds. Glycogen is similar to amylopectin found in plant, except that it is more highly branched. The branches occur between 12 to 14 glucose mono 12 to 14 glucose units. The branches serve to increase the solubility of glycogen and make its sugar unit accessible. When there's a need for energy in the body, glycogen is broken down. The glucose released goes through the glycolytic pathway and is used to generate energy. Starch, on the other hand, is a nutritional reservoir of polysaccharides in plants. Example, you find starch in potatoes and tubers of yams. It is reserved carbohydrate for the development of a developing plant. Amylose is essentially considered as a linear polymer of starch. Amylose consists of glucose residue in the alpha 1,4 linkage. Amylopectin is a branch form of starch and has about 1 alpha 1,6 linkage per 30 of alpha 1,4 linkages. And so it is like glycogen, except that in branching, it has a lower degree of branching. Amylose and amylopectin do not exist free in nature, but they occur as crystalline aggregates called starch granules. Now, this is the structure of amylose. It has a non-reducing end and a reducing end, and its sugar molecule linked in the alpha 1,4 linkage. And for amylopectin, there is always branching at alpha 1,6 linkage. So both amylose and amylopectin they are found in starch. They are the forms in which starch exists in the crystalline form. Cellulose is the most abundant organic compound that is found in the biosphere. And cellulose is a linear condensation polymer of glucose having beta 1,4 glycosidic linkage. And the simplest repeating unit of cellulose is 2 beta-D-glucose linked by beta 1,4 glycosidic linkage that is called cellulose. The beta configuration allows cellulose to form very long straight chain that is found in cell walls of a plant. Heteropolysaccharides, examples are the Glycosaminoglycans. Higher uronic acid is a repeating disaccharide unit of glucuronic acid and N-acetyl glucosamine, which are linked together by alpha 1,3 linkage. Higher uronate is an essential component of the extracellular matrix of cartilage and tendons, to which it contributes tensile strength and elasticity as a result of a strong interaction with other components of the matrix. Chondroitin sulfate also is a repeating disaccharide unit of glucuronic acid and N-acetyl galactophosphate. Chondroitin sulfate contributes to the tensile strength of cartilage, tendons, ligaments, and the walls of the aorta. Now, this is the structure of some glycosaminoglycan repeating disaccharide units that are found in the higher uronate. We have the N-acetylglucosamine interacting with the glucuronic acid, and in chondroitin 4-sulfate, glucuronic acid reacting with N-acetylgalactosamine 4-sulfate. These are the repeating structures that make up these molecules. And with this, we have come to the end of the class. I hope to see you next time. Do have a great day. Thank you for good listening.